Okay, let's unpack this. Welcome to the deep dive, where we try to cut through the noise and get straight to the knowledge you need. Today, we're embarking on a pretty fascinating deep dive into one of your body's most critical, yet, often unheralded defense mechanisms. The complement system, it's complex, it's crucial, part of our immunity, but with some really surprising functions. It kind of stands at this intersection of protection and, well, sometimes peril. Our mission today, to give you a genuine shortcut to being well-informed about this incredible system. We'll get into its key components like the central players, C3 and C4, understand the lightning-fast cascade they kick off, and then reveal how this powerful protective squad can sometimes have this double-edged effect, especially in sensitive organs, maybe leading to conditions like glomerulonephritis in the kidney.
And that's really where the aha moments lie, I think, in understanding this system. It provides such a clear, almost, well, poetic illustration of the intricate balance needed for the body's defense. And when that delicate equilibrium is disrupted, maybe even by a system designed specifically to protect us, the consequences, they can be profound. It truly makes you appreciate the inherent complexity and, the tightrope walk our immune system performs every single day.
Right, so here's where it gets really interesting. Imagine the complement system as your body's most seasoned rapid response team,
Yes.
right? A frontline defense that's always on duty. We're talking about a vital system of serum proteins, basically. Proteins always floating around in your blood that are deeply involved in inflammation and immunity.
Yeah.
What's key here, and I think often overlooked, is that it's a core part of your innate immune response.
Exactly.
Innate, which means it's not waiting to be trained or, specifically recognize a new threat like your adaptive immune system does. No, it's always present, always patrolling, ready for action, like instantly.
Yeah.
It's the ultimate first responder.
That is a perfect way to put it. Yeah. Think of it less like a maybe specialized SWAT team and more like the city's entire emergency services system, police, fire, paramedics, all on constant patrol, just ready to deploy. Its core functions are incredibly diverse and potent. It mediates activities that include activation of phagocytes. Those are the immune cells that literally eat or engulf foreign invaders.
The Pac-Man of the immune system.
Kind of, yeah. It can also perform direct cytolysis of target cells, which basically means it punches holes directly into bacterial or infected cells, causes them to burst.
Wow.
And critically, it helps with the coding or opsonization of microorganisms for uptake by cells expressing complement receptors.
Opsonization, that's like flagging them, making them obvious.
Precisely. It tags invaders, makes them far more visible, more palatable, really, for those hungry phagocytes to grab onto and consume.
Yeah.
So it's this multi-talented defense squad, always on standby, ready to identify, neutralize, and clear threats.
So it's like the body's emergency services, constantly circulating, constantly vigilant, and able to kick off a rapid cascading response before a specific antibody even gets a chance to form. That's fast.
Incredibly fast.
But how does this intricate system actually work? I mean,
Yeah.
how does it get activated? And what are these serum proteins, especially as central players C3 and C4, what are they actually doing to orchestrate this whole defense?
But what's truly fascinating here is that the complement system operates through an incredibly efficient enzymatic cascade.
Enzymatic cascade.
Like domino. Exactly like dominoes. Imagine a meticulously set up line of them, but with each falling domino not just knocking over the next one, but also triggering multiple others, amplifying the signal exponentially as it goes.
Ah, okay. Amplification.
Yeah.
Yeah.
And this cascade, it can be initiated in a couple of key ways, which shows its versatility, either through reactivity with bacterial factors directly, that's the alternative pathway, or with antibodies that have already latched onto a pathogen, that's the classical pathway. So this initial spark, whether it's direct pathogen recognition or an antibody tag, it sets off this chain reaction. One protein activates the next, which activates the next, amplifying the signal with remarkable speed and precision. And this cascade, it produces a whole host of important breakdown products. These are essentially active fragments of the original complement proteins.
Fragments doing the work.
Exactly. And these fragments are recognized by specific receptors on the surface of various immune cells, and that leads to cellular activation and enhanced function. We're talking about, well, a whole alphabet soup of fragments here. C through, C3B, C3B, C3A, C4A, and C5A.
Yeah.
And while they all play roles, C3 is often considered the real linchpin, the central hub of this system.
Why C3 specifically?
Because its activation is pivotal for all the main complement pathways. Its breakdown product, C3b, is particularly critical. It's the main tag marking targets for destruction.
Okay, C3b is the tagger. But you mentioned other C3A, C4A, C5A. Let's zoom in on those for a second. These aren't just random codes, right? They're doing something important.
Oh, absolutely. They are incredibly potent chemical signals. These fragments function as anaphylatoxins.
Anaphylatoxins. Sounds serious. Like anaphylactic shock.
Photolylated, yes. It means they can trigger acute inflammatory responses, redness, swelling, heat. They get things stirred up.
Okay.
They're also powerful chemotactic factors.
Chemotactic, meaning they attract things.
Exactly. They're like chemical magnets or maybe flares, calling other immune cells neutrophils, monocytes rushing to the site of infection or injury. Think of C3A and C5A as those flares fired into the sky, signaling reinforcements needed now.
Got it. The distress signals.
Right. And then C3b, as we said, acts as that potent opsonin, effectively sugar-coating pathogens to make them much more appetizing and recognizable for phagocytic cells.
Makes them easier to eat.
Much easier.
Yeah.
So you can see how C3 and C4, through these various breakdown products, the A fragments calling for help, the B fragments tagging the target, they're truly central to kickstarting and orchestrating this entire defensive cascade. It ensures rapid, effective clearance of threats.
Okay, that makes sense. It's a powerful, coordinated response.
Right.
But now for the surprising twist, the almost paradoxical downside you mentioned. So what happens when this incredibly powerful protective system, doing all these amazing things with C3, C5A, what happens when it sometimes goes, well, wrong? You said it can actually contribute significantly to tissue damage. That seems completely counterintuitive for a system meant to defend us.
It does seem counterintuitive, and this raises an extremely important question for us. How does a system so meticulously designed for protection end up causing harm? Harm to the very body it's supposed to defend. A prime and often quite severe example of this paradox is found in something called type 3 hypersensitivity.
Type 3 hypersensitivity. Okay.
In this scenario, the issue starts with these things called soluble antigen-antibody immune complexes.
Soluble antigen immune complexes. Bit of a mouthful.
It is. Picture tiny sticky bundles. They form when antibodies bind to foreign material antigens. But instead of being cleared out efficiently, these bundles remain floating in the bloodstream.
Okay, so debris that hasn't been cleaned up properly.
Sort of, yeah. And these complexes then incorrectly, or maybe overenthusiastically, activate the complement system.
So complement gets triggered by our own antibody complexes, not just pathogen.
Exactly. And the critical consequence here, this is really key, is that these immune complexes, once they've been activated by complement, they can become deposited on the membrane surfaces of various organs.
Deposited, like stuck there.
Stuck there. We're talking about vital places like the kidney, lung, and synovium, that's the lining of your joints.
Oh, wow. Not good places.
Definitely not. These complexes accumulate in these sensitive tissues, essentially turning these formerly protective elements into targets for a subsequent immune attack. In the kidney, for instance, these deposits often lodge in the delicate filtering units, the glomeruli. This causes a condition known as glomerulonephritis.
Glomerulonephritis, right? You mentioned that. And that can severely impair kidney function.
Severely, yes.
So explain the mechanism of injury for us. How does the damage actually happen once those complexes are stuck there and complement is activated?
Okay, so once these immune complexes are deposited and the complement system is activated locally, right there in the tissue, the byproducts of complement activation, specifically C3A and C5A, those flares we talked about.
The chemotactic factors.
Precisely. They act as powerful chemotaxins for acute inflammatory cells. They send out that irresistible beacon.
Calling in the troops.
Calling in the troops. Hordes of cells, particularly polymorphonuclear cells or PMNs, your neutrophils are drawn to the area where those complexes are deposited.
Neutrophils? Aren't they usually good guys?
Yeah.
Fighting infection.
They are vital for fighting infection, absolutely. But think of them as cellular grenades. When they arrive in massive numbers in response to these deposited complexes, especially in delicate places like the kidney's glomeruli, their intense infiltration into the tissue then causes direct tissue injury. Why? Because they release their destructive lysosomal enzymes somewhat indiscriminately.
Friendly fire. Or maybe not so friendly.
It's collateral damage, essentially. An immune civil war where the surrounding tissue gets caught in the crossfire.
Leading to inflammation and damage in the kidney, the lung, the joints.
Exactly. And to add another layer to that pathology, what we often see in these situations is that platelet aggregation occurs.
Platelets get involved too.
Yeah.
Clotting cells.
Yes. This results in microthrombus formation in the vasculature within these affected organs. Tiny blood clots form in the small blood vessels.
Which must make things even worse.
It absolutely does. These tiny clots further obstruct blood flow and oxygen delivery, exacerbating the damage in an already vulnerable tissue like the kidney glomerulus. It can lead to this vicious cycle of inflammation, clotting, and tissue destruction that can be very, very difficult to break.
Wow.
And that's how the very system meant to clear threats can, when it's misdirected by these immune complexes, inadvertently create the conditions for severe disease like glomerulonephritis.
It's an unexpected truth, really. A system meant to defend, with its C3 and C4 components kicking off such a vital cascade, can, under specific conditions, inadvertently attack, leading to significant harm in critical organs. Like the kidney, where it causes glomerulonephritis. It just transforms from guardian to aggressor. It really highlights the fine line our immune system walks. Okay, and that, I think, wraps up our deep dive into the fascinating and, yeah, sometimes frightening world of the complement system. We've explored its essential role as a rapid frontline defender. We dug into the specific actions of key components like C3 and C4, and we uncovered the really surprising way it can contribute to severe tissue damage, like glomerulonephritis, when these immune complexes get deposited in organs like the kidney, lung, and synovium.
And if we connect this to the bigger picture, I think the critical takeaway is this. Your body's immune system is just a masterclass in balance, in regulation. Even its most vital protective mechanisms, like the complement system, which is constantly scanning for threats, it requires incredibly precise control, tight regulation, just to prevent it from becoming an agent of disease itself. It's really a testament to the delicate equilibrium that most of the time keeps us healthy, and it shows the profound consequences when that balance is lost.
Yeah. So as you go about your day, maybe consider this. How many other seemingly protective mechanisms in the body might also have a hidden double-edged nature, constantly navigating that razor-thin line between defense and potential self-harm? It makes you think differently about how we understand health, disease, and perhaps even how we approach therapies that might, tip that delicate balance. What else in our bodies are we relying on that could, maybe under the wrong circumstances, turn against us? Something to think about.